Hi, everyone, and welcome to Drug Discovery Day with Endpoints News. Thank you for joining us, and thank you to Emulate for sponsoring today's event. My name is Sarani Fernando, and I'll be moderating a few panels today, starting with our keynote lecture. We'll first hear from David Walt, Professor of Biologically Inspired Engineering at Harvard Medical School. Then we'll hear from Carly Strelez, Manager of the Organ on a Chip team at Ellison Institute of Technology. After this panel, join Endpoints News' Ryan Cross for a fireside chat with Stuart Schreiber from Arena Bioworks. Then we'll continue after that with two more thought-provoking sessions with some top industry speakers. The first on improving efficiency of drug discovery, and the second on gaining human-relevant insights into infectious disease. If you have any questions today, please feel free to submit them with the Q&A button on the bottom of your screen, and the team at Emulate will be happy to follow up on questions after today's event. Also, just to add that all of today's sessions have been recorded and will be available on demand starting April 5th. And with that, I'll welcome David Walt to kick us off. David?
Thank you, Sarani. Okay, well, today I have the privilege of kicking us off on a topic that's near and dear to my heart, transforming drug discovery with next-gen biotechnology tools. And this topic really, I think, I was invited because I'm involved with a number of tools companies in this space, and I'll be covering a number of them. So I just want to make sure that I disclose that I do have relationships with these companies, serving either as founder, director, equity holder. But again, I'm not trying to push any of them. I'm just trying to use these companies' technologies as examples of the space in which we're going to describe today in drug discovery tools. So the outline here is really to start with sort of an older technology with next-gen sequencing, but also show how by combining it with AI, it enables high-throughput discovery both of proteins for antibodies as well as for enzyme discovery. We'll then move to a relatively new but extremely exciting area called spatial biology, and then we'll wrap up with better tools for predicting drug responses, both with respect to organs on chips, which I know Carly's going to talk about a bit in the next talk, as well as with ultrasensitive protein detection. And so I want to start out by just setting the stage here. The progress in the ability to analyze the human genome is really unprecedented. We went from the first human genome project, which developed the first human genome in roughly the turn of the century, 2000, 2001, at a cost of $3.8 billion. It involved thousands of individuals, took 13 years, and it involved 40 different institutions. Today, we know that the NovaSeq instrument that's put out by Illumina, but also other instruments that are similar in terms of sequencing by synthesis, can do terabytes of sequence per day at a cost of $200 per genome. And the accuracy of these genomes is much higher than what was developed over the first 13 years of the Human Genome Project. These also take very few manual inputs, so less than 1 FTE is required to run one of these instruments. And so, just to put this in perspective, I think many of you have seen this kind of a curve, that was put out by NIH up until 2021. They stopped putting it out, but what we see today is that, compared to the end of the Human Genome Project, we can complete the entire Human Genome Project on one of these high-throughput instruments in 7 minutes. So entire high-quality human genome every 7 minutes. It's 19 million times cheaper, and one instrument is 1 million times faster than 40 labs combined. And so, you can see just not only by the deviation from the Moore's law line here, but also just, this is just an incredible advancement in technology over a very short period of time. And so I'm going to go through a couple of the technologies that are enabled both by the high throughput sequencing capability, as well as by coupling that with the sophistication of classification algorithms, also known as AI today. First is a company called Protillion Biosciences, and what they've done is, just as a DNA sequencer outputs tremendous amounts of DNA sequence data, the Protillion platform has developed an automated chip-based instrumentation that's actually built on the DNA sequencer technology. And what it does is it enables that instrument to produce massive amounts of quantitative protein binding data across an entire library of antibody fragments. It generates the largest data sets of protein binding for machine learning, and it can generate up to 1 million equilibrium binding curves simultaneously, and can also look at the kinetic parameters of those binding events. And so, just to sort of take you briefly through the technology, this is built on a sequencing platform. So what is done is genes of interest are hybridized to regions on the sequencing flow cell. So you can see that over on the left-hand side. There's an amplification step called bridge amplification. That's a PCR step to amplify those single strands of DNA into clusters. And then what Protillion does is it then uses in vitro transcription and translation such that each of these individual sequences now gets translated and a protein gets expressed at the end of those sequences, so that you now have proteins that are being expressed. And these can be peptides, they can be antibodies, they can be pretty much user defined. So the advantage of this is that instead of winnowing candidates where every candidate in each screen gets eliminated during the selection process, you can just continue to look at data from all members of the library, and there's again millions and millions of proteins being expressed on this kind of a chip. And so whereas existing discovery methods use sort of a sequential destructive kind of platform. That is, they will look at single cells, they'll look at a phage display, they'll look at hybridomas, they'll select those antibodies, for example, that come out, they'll express and purify them one at a time, and then they'll measure those binding. In contrast, with this really high throughput technology that is now available on a platform like this one, one can now simultaneously collect millions of binding events simultaneously, and it's in a non-destructive manner. That is, once you wash that, you can then interrogate things simultaneously or sequentially without destroying the proteins that are presented on the platform. And so you can interrogate it under different conditions of pH to look at how binding is affected by pH, so that you can optimize it for a particular delivery in a particular organ. You can screen for multi-target specificity if you want to have multiple things recognized or not recognized, and you can do this on an affinity screening basis as well, so that you can screen for your ultra-high affinity binders. So I'm going to move to another platform. This is one that's been developed by a company called Arbor Biotech, and this is really looking at high throughput enzyme discovery. And so what the company does here is they use a discovery platform that takes advantage of the huge metagenomic database that exists from microbial sequencing. These are both public databases as well as customized private databases that they've sourced from nature. And what they can do is they can annotate those using various annotation tools, and they've annotated more than 3 billion unique protein sequences to date. What is involved in the process at Arbor is that this screening process, as you can see sort of in the second box here, is first done, then proteins are expressed and validated to see if they actually have the activity that they're screening for. One can then learn from it, again, search for additional candidates that might have better properties. And then you build sort of a taxonomy of different members of families that have the ability to express those particular activities. And then once you've identified those, you can then use additional algorithms to help you design and engineer and optimize those activities. And so, for example, some of the candidates that have been identified just through this initial screen can then be optimized through site-directed mutagenesis or directed evolution. So there's an incredible scale of experiments in design that are enabled by this, not only in terms of identifying lots and lots of different protein sequences, but the screening, the high throughput screening has gone up in terms of its capability such that now they can do wet chemistry or wet biochemistry on over 300,000 cell experiments per year. And the time to go from a screen to a lead has decreased by roughly tenfold with roughly 10 terabases in the data set. This is just really to sort of wrap up and show you examples of discoveries that were made specifically in the nuclease space here. So this is, these are analogs of Cas9 that are used for CRISPR. And you can see that there's a wide variety of different nucleases, different sizes, which mean different ways that these can be delivered. The guide RNA length that these recognize or require can be very small, from 42 nucleotides all the way to 158. So you can get exquisite specificity. And as you can see, they derive from a wide variety of different metagenomes, groundwater, lakes, oceans, etc. So switching to spatial biology, excuse me, now, here, this is really, I think, a very exciting area. There's, a number of commercial entities that are exploring this space. The one I'm presenting here is a company called VizGen, which was, came out of the lab of Xiaowei Zhuang at Harvard. And, you can think of this at multiple scales. A mouse brain, for example, contains hundreds of thousands of cells, but what you'd really like to be able to look at is gene expression in those cells, and this technology enables you to do that for billions of transcripts in individual cells and even subcellular environments. And so you can see all the way on the right-hand side here, there's countless analytical data points. This is within an individual cell. You can see many, many different genes being expressed. And so this is really enabling drug discovery in significant ways. For example, with lung cancer, human heart, infectious disease, and gene therapy, there's really three, I think, major applications here. One is you can discover new targets by characterizing the biological functions of different cell types in those tissues and basing those cell characterizations not on phenotype, but on genotype, which is much more molecular in nature. You can do biomarker discovery and validation that can help you stratify patient cohorts and predict their therapeutic effectiveness based on the way that these genes present in the particular tissues. And then there's also just being able to confirm the underlying mechanisms of action of drugs as you begin to look at how cell and gene expression levels change in the presence of the drugs. Here, is just one example of where spatial biology provides new information for drug discovery, and that's the tumor microenvironment. And you can see that there's spatial relationships between the cancer tissue and immune cells, and those relationships, as many of you are aware, will really be predictive of whether a patient is going to respond to a drug or not, based on whether there's infiltrated tumor that's inflamed or whether it's exhausted, and the type of drug that one would use in each of these different tissue and cancer types would be different depending on how the relationship between these cells presents itself. And so, I think the other area where spatial biology has probably had its biggest impact so far, at least in the basic research space, is in the brain cell map, which this is an example of a paper that was presented by the Allen Institute, and these were really fundamental studies that looked at entire brains, entire mouse brains, to look for to build a massive brain cell map that could identify potential new targets for Alzheimer's drugs, and of course, other neurodegenerative disease targets could be identified as well. So, again, this is just one example of where this kind of technology can be used on the front end for discovery of new targets. So, our sponsor today, Emulate, provided me with these slides as well. This technology came out of the Wyss Institute at Harvard University. I'm actually here in my office here today, and this was originally developed at the Wyss by Don Ingber's group here with the publication in 2010 of the first lung on a chip. And I'm not going to go through this in great detail, but the way that Don and his team worked here is to develop both cell layers of both epithelial cells and endothelial cells here, and then very cleverly recognize that mechanical stimulation that's akin to the breathing mechanism of the lung, that is the expansion and contraction of the lung, that mechanical the mechanical stimulation was essential to really recapitulating the activities of the cells that were in that lung on a chip, and that really, I think, transformed the field in organ on chips. And so, with these organ on chips, they deliver a greater human relevance because they're using human tissues. Obviously, we'd like to get away from animal models, but many of the animal models, of course, they fail in late stage studies because humans and animals are not the same. So the question becomes how to use these organs on chips through the drug development pipeline. And so when we look at their applications, we can see multiple benefits across really the entire pipeline of drug discovery. In the early discovery phase, the chips can be used to develop disease models with closer to human gene expression in order to identify and validate drug targets. In the lead optimization phase, they can be used to study the candidate efficacies of different drug candidates in order to rank and order and optimize those lead candidates. And then in the preclinical safety phase, they can be used as a valuable screening tool to predict different mechanisms of unexpected toxicity. And this is, I think, one of the areas that they're used most commonly, as well as assess the human relevance of the toxicity that may have been observed in the preclinical animal studies. And then finally, they can even benefit the clinical trial phase. For example, when drug candidates enter the phase 1 and 2 trials and produce unexpected safety signals, that you can reflex to organ-on-a-chips to really understand, why did this exhibit that particular untoward effect. So, this is just a recent example of a company who is implementing this kind of a scheme, that licensed the technology and is using it for detecting inflammatory lung diseases, including COVID-19. This really, I think, highlights, I would say, one of the major uses of these organs on chips, and that's the liver chip model, to really screen for toxicity. In this particular study that was carried out by Emulate, the 870 human liver chips were analyzed. It was able to correctly identify 87% of the drugs that cause drug-induced liver injury to patients, despite passing through the animal testing. And it replicated the key histological structures and functions of the liver. It distinguished between 7 pairs of small molecule drugs and their non-toxic structural analogs, and it predicted the clinical responses of 6 additional selected drugs. Again, you can see 870 chips were analyzed, which really underscores that the system is robust and easy to use platform. So, I'm not going to, go through this in great detail, but, these have been used, over the last, 5 years or so, presented in many, scientific and also, public interest journals, just showing the impact that these have had, through intestine chip, liver chip, lung chip, and other applications. And of course, look, there's no doubt that animal models, and I'm sure we'll hear this in the next talk, have contributed to major advances in medicine, but they also aren't great necessarily at predicting and recapitulating human response. And so this organ-on-chip technology allows researchers to recreate the human microenvironment, and they can also provide an alternative to animal testing, although initially we probably will understand that we're still going to have to rely on those animal models, at least in the initial studies, until these technologies are further validated. So, the last topic I wanted to cover today is liquid biopsies, where these have been used in all sorts of ways for both detecting early stage cancer, but also for being able to predict therapeutic responses. I would say that the biggest issue with these liquid biopsies today, in my opinion, is that they rely primarily on genomic analysis. And so I think there is now, in the context of this talk today, there's really a serious recognition that we can't simply rely on genomics anymore. We have to begin to look at beyond genomics to understand human biology, including obviously the messenger RNAs, which is genomics, but really understanding them both spatially and temporally through the spatial biology platform that I described earlier, that the proteins that are translated are important. So that's the proteomics, the processes of gene expression, that's the epigenomics, the state of the cell, which is the metabolomics, and the state of chromosomes, which in some cases, particularly with cancers, can be detected through what are called fragmentomics, and of course the trillions of microbes that we live with with the microbiome. And so I'm going to focus on one technology that was developed out of my laboratory called Simoa, single molecule arrays. This is a digitized immunoassay that really allows you to literally count protein molecules. Again, I'm not going to talk in detail about the technology. This technology is commercialized by a company outside of Boston called Quanterix, public company. There's about 1,000 instruments that have been placed worldwide. They have service businesses where they have run over 2,000 studies for pharma and researchers. They've discovered 550 new proteins that are below the detection limit of conventional immunoassay technologies, and lots of publications have come out. And so really, the enabler here is the fact that you can look under the surface of what the traditional ELISA immunoassays have been able to access for the last 40 years, such that we can go 1,000 to 10,000 times more sensitive than that technology, and that opens up the ability to detect thousands of additional proteins. And so, what this enables with respect to drug discovery is really detecting disease at a very early stage, where we know that early detection predicts better outcomes. So better prognosis is a consequence of early intervention due to the rather lower disease burden in patients when they're detected earlier. Clearly, the same technology can be used for looking at residual disease or therapeutic efficacy, but again, a whole spectrum of things that these ultrasensitive protein assays can be used for. And examples here where these have already entered into either research or preclinical, and in some cases clinical phase, include neurodegenerative disease, cancer, lots of inflammatory diseases, as well as infectious diseases, with a whole host of new biomarkers that have been discovered. These are the technology is being used significantly in the neurodegenerative disease arena, primarily for Alzheimer's, but also in Parkinson's and some other neurological diseases. You can see, some of the clinical studies that are being done. And, just to highlight one of them, this particular marker, this phosphatau 181, is actually used on the Leqembi label. That's one of the drugs for Alzheimer's disease and so this is on the label as a biomarker that really is only detectable at this ultrasensitive level. And so, what I've hoped to do in this half hour is really show you a spectrum of tools that can be used, from new sequencing capabilities to spatial biology, to new ultrasensitive methods for detecting proteins and other molecules, but also the ability to use the organ-on-chip technology to really predict disease drug efficacy, and also look at toxicity. So again, we're not answering questions here, but feel free to pass those on to the organizers, and we'll try to get back to you. Thanks very much.
Great. Thank you, David. Thanks for that really insightful presentation. Such an evolution in this area of drug discovery. So thanks for that. And now please welcome our next speaker, Carly Strelez. Carly?
Great. Thank you very much for the opportunity to share our research. I'm at the Ellison Institute of Technology, managing a team that's been working with organ-on-chips for many years. And I'm going to be talking today about how we're using a patient-derived organoid model system to model the colorectal cancer microenvironment, and hopefully to better understand cancer progression. These are my disclosures. We are focused mainly on the colon cancer space, although we do branch out every once in a while. We're really focused on understanding the tumor microenvironment, all the different cell types that are present, whether it be immune cells to the other stromal components like the cancer-associated fibroblasts, the endothelial cells, Cancer is really a complex system, and therefore we do need better preclinical models to really recapitulate aspects of the complex tumor microenvironment, increase the mechanistic understanding of cancer biology, and of course, study drug response and hopefully identify novel targets. We have been working with the Amulet organ on a chip, which had a great introduction in the previous talk. We do have a 2-channel system that we work with a top epithelial channel, a bottom endothelial channel separated by a porous membrane. One of the key factors that was also highlighted in the previous talk are the mechanical forces that are present in this Amulet model system, not only the fluid flow, but also the mechanical stretching of the chambers and the cell culture area to actually mimic. In the previous talk, it was breathing motions, but in this particular case, peristalsis, which we've been working with extensively to understand how the colon cancer actually deals with the mechanical signaling. I won't be talking about that too much today, but I did want to highlight the importance of the mechanical forces and how in all of the studies following, we do use the mechanical properties of the chip to mimic peristalsis. We've worked with various different cell types and layering them on to try to mimic and recreate some aspects of the tumor microenvironment. We've worked extensively with colon cancer tumor cell lines. We've worked our way up with cancer-associated fibroblasts for starting to look at the immune cell populations. And what we've traditionally done with this organ on a chip is really try to understand the functional readout that's happening. And the one that we're particularly interested in is understanding invasion, the early invasive events of tumor cells migrating or invading into the bottom endothelial channel. And as you can see here, we do a lot of fluorescence microscopy to understand how green tumor cells that are seated in the top channel then invade through the ECM layer, through the porous membrane, into the endothelial cells and start to colonize. Specifically, what I'm going to be talking about today is a lot of work that we've done over many years to try to build out our patient-derived organoid on a chip model systems. And so what we've been able to do is take 6 patients. So these tumors were derived from a variety of different stages. Some of them have different KRAS mutations. Some of them are wild type for this particular mutation, different colon tumor locations as well. And what we can actually do is we can derive the organoids from this patient tissue, and then we can put them onto the top channel of the organ on a chip model system. And so for our first pilot experiment, our proof of concept experiment, what we were able to do is we were able to sequence at various time points of this model development. So taking the tumor tissue, taking the organoids from which they were derived, and then after the experimental conditions and performing RNA-seq at those various time points to understand what added value are we getting from these organ on chips. We can also, because this is a microfluidic device, we can also sample the effluent that is collected and perform various effluent analysis. In this particular experiment, it was a metabolomics experiment, but great ways to look at various biomarkers potentially, or other things that are being secreted or taken up in the medium. And as you can see here, what we started to see as we put all of these organoids on a chip is that we do see quite a bit of patient heterogeneity, even though all of them do recreate the barrier that is very important within the colon, as well as the various patient heterogeneity seen with our RNA sequencing data. Furthermore, when we went to compare across model systems, so again, what I'm looking at here is RNA sequencing data of the organoids on the chip, and then the tumor. So this is all patient matched. When we started to look at the differentially expressed genes across these different comparisons, you can see that the chip, as compared to the tumor, had less differentially expressed genes than potentially organoids versus tumor. And so given that there's potentially a closer transcriptomic similarity to the patient tumors, we wanted to see how can we start to use these tumors to the organoid chips to understand response to therapy and potentially try to develop novel targets. What we've been working on recently is trying to look at the response of these organoids to chemotherapies that are clinically relevant. So this is your, within the colon space, this is 5-FU, this is irinotecan, this is cetuximab. And what we're able to do is we're able to dose these organoids, the patient-derived organoids on chip. We're able to do traditional viability measurements, such as cell titer glow, for example. We're always able to sample the effluent and do downstream analyses, such as mass spec or other proteinomic quantifications. And again, we're very interested also in the invasive capabilities of these organoids. And so we're able to also do the imaging based analysis of how these organoids are invading into the endothelial channel as a way of measuring cancer progression in the space of these, in the context of these chemotherapies. And we're just starting to understand what additional information are these organ on chips giving us with the response to chemotherapy, and how can we compare back to the organoids in a 3D culture setting? So the traditional drug screening in organoids, and how does that match, or what added information can we get from on chip? And hopefully that data will be coming out soon. So this is an example of how we're looking at common chemotherapies. And I wanted to transition over to an example of how we're using these chips to understand more specific signaling differences that we can see in the tumor microenvironment. There's been a lot of studies coming out recently looking at the connection between the nervous system and cancer, the neural signaling that's happening in a variety of cancers, not just brain cancers. And within the space of the colon, there's been a lot of studies starting to understand what's happening when the cancer cells are present in a very neurotransmitter-rich environment, like GABA, for example, serotonin, acetylcholine. And so not only do these neurotransmitters exist within the context of the tumor microenvironment, there's also significant effects on the other cell types, such as the endothelial cells, the immune cells. And so we're really focusing in on this particular section of the talk of understanding what is going on between the neurotransmitters and the tumor cells. We looked at the tumors in our biobank. So this is across 10 different tumors. This is the tumor tissue. And we were able to see here that some of these neurotransmitter genes, so this is looking at a neurotransmitter panel across our tumors, there are several genes that are actually quite highly expressed within the tumors that we wouldn't necessarily have expected potentially. And you can see that several of these genes are within the GABAergic signaling pathways. So what we also did is we took the inlet and outlet media, the effluent in particular, from the top and bottom channel, did perform some metabolite extraction and then some mass spec-based metabolomics to understand what is happening with our various organoid on a chip. But then also we specifically looked at a section of neurotransmitters, a selection of neurotransmitters across the different patients. And as you can see here, we're looking at 5 different organoids on a chip here, and you can see that there's quite a variety and quite heterogeneous amount of these neurotransmitters present in the effluent. For example, serotonin, this UK organoid line, as we go from day 0 of the experiment all the way to the end of the day 6 of the experiment, you can see that there's quite a bit more serotonin present in the effluent for this particular organoid chip, which may not be the case for other organoids. GABA seems to be of interest for us. And so we specifically looked at GABA and saw this particular US line here, organoid line, had quite a bit more GABA present than the other organoids. And specifically, as we look at from over the time course of the experiment from day 0 to day 6, a significant increase in the amount of GABA present. And so we decided to focus our continued efforts to understand what was happening in a GABAergic environment with these particular 2 organoid lines, the US and the UP organoids. We did validate that on chip, performing some immunofluorescence staining of GABA on chip for these 2 different organoids. So we have our GFP labeled organoids here for the US patient and the UP patient, and you can see that there is more GABA present on chip with the US, confirming our effluent analysis. And we also went one step further, looking at the actual tumor tissue and taking the performing immunofluorescence on the tumor tissue slides. And you can see here that there's more magenta, so there's more purple overlapping with the tumor cell here in the US patient, indicating that there is more GABA present in the tumor microenvironment as compared to the UPs. So this is a nice, very small, especially when we're thinking about drug discovery, small numbers here, but a highlight of how we can start to use the organ on chip to recreate some of this neurotransmitter-rich GABAergic tumor microenvironment. We went back to our cell lines to do some follow-up mechanistic experiments to try to understand what was going on with this GABAergic environment. Again, we do a lot of our work trying to understand how these tumor cells invade into the endothelial layer and colonize. And so as you can see with our HCT116 tumor cell lines, as you increase the amount of GABA, you see more cells invaded into the endothelial compartment, giving us a sense of this GABAergic process might be increasing invasion, which could be potentially worse for the cancer progression. And we wanted to know, since there is a lot of GABA present, what is that actually? What are the signaling pathways that are potentially implicated? There's a lot of different options, especially within the tumor cell area. And so what we actually did is we introduced labeled GABA into the system exogenously and then harvested the cells. And this is measuring the amount of GABA that's actually intracellular in the HCT116 tumor cells. And as you can see, when we add GABA exogenously into the system, we do see GABA intracellularly, indicating that for this particular experiment, tumor cells are actually taking up GABA to do their signaling process. And so we wanted to add a follow-up to understand if we can target the GABA signaling to block tumor cell invasion in this particular model system. And one of the ways that cells can utilize intracellular GABA is they can metabolize it and using this ABAT enzyme to make succinic semialdehyde and into the TCA cycle as an energy source. Vigabatrin is a particular drug that will block the ABAT enzyme. And so what we did is we added vigabatrin into our system, and you can see here with two different colon cancer cell lines, we do see different responses to this vigabatrin. The HCT116s, the tumor cell line that I've been working with in the past two slides, did show increased invasion in the presence of vigabatrin, whereas the DLD ones did not show a similar response to vigabatrin. Then we move over into the organoid space. This is the patient-derived organoids. And for two different organoid lines, we do see different responses with how these organoids are responding to ABAT inhibition. The US line decreases invasion, whereas the UPs do not. We're still understanding what this all means, but you can start to see how these chips are useful in understanding some of the patient-level differences that exist within the tumor microenvironment and giving us an opportunity to target those differences. And so just to wrap up quickly, again, I can't highlight enough how important the mechanical forces are in the tumor microenvironment, specifically in the colon cancer. So we have developed a colon cancer on a chip with patient-derived cells that has mechanical forces to recapitulate important aspects of the tumor microenvironment. And we do think that some of the GABAergic signaling that has been understudied can result in increased invasion in certain environments. And we're hoping to do more follow-up studies to understand what the certain microenvironments means. I did highlight the great work of my organ on a chip team in past slides. We have a great team. This is a full collaborative environment at the Elson Institute of Technology across various teams, the molecular analytics team, as well as the cell line team. And then, of course, this work was funded by several grants that were awarded to Dr. Shannon Mumenthaler. And we would not be where we are able to do with any of this work without the patient tissue. So thank you very much.
Great. Thank you, Carly. Incredible stuff. And thank you again, David. Both really great and insightful presentations. So coming up in 5 short minutes, Ryan Cross will be joined by Stuart Schreiber, founding CEO at Arena Bioworks, for a fireside chat. And in the meantime, please feel free to answer a brief audience poll that will come up in just a moment. We'll be right back.